Morning, everybody. Today's topic is cell cycle. Growth and development. Every organism depends exclusively on multiplication and enlargement of its cells. All the cells of the body arise from the division of the pre-existing cells was emphasized by Rodolph Virchow in 1858. The development of the multicellular organisms from unicellular zygote was achieved by cell division, growth, and differentiation. Cell division is the mode of cell reproduction. It's nothing but the multiplying. No, multiplying means reproduced. That's all. It provides the basis for one form of growth for both sexual and asexual reproduction and for transmission of the heredity from one generation to another generation. It has the perpetuation to one generation to other generation from the genetic material only, no? The division of nuclear cells is achieved by two integral activity such as nuclear division, otherwise called as karyokinesis, reported by Slaker, and division of cytoplasm reported by Whiteman. Both are reported by the 1887, same year, karyokinesis and cytokinesis. Karyokinesis followed by the cytokinesis, but sometimes does not results in formation of the multinucleate cells. Now question arises as to why and how cell divides. Answer of why: there exists definite ratio between the cytoplasmic and nuclear volumes of the cell. It is referred to as a plasmic index. As and when this ratio is disturbed, the cell tend to divide. How? The answer of how has been discussed in the following. Importance of the cell division can be appreciated by realizing that in higher eukaryotes, life starts from the formation of zygote. Zygote is nothing but fertilized egg. No? From the single cell, trillions of cells developed by successive divisions. These cells undergo the process of differentiation and various organs or formation takes place. During this, the rate of cell division and number of cells vary. In a fully formed organisms, there are fewer dividing cells as compared to the during the course of development. However, in adult tissues, bone marrow and germinal tissues, in plants, meristematic tissues, cell division takes place continuously. Continuously growing, no? At the apical regions where cutting means continuously growing, no? That is naturally occur. The somatic cells are body cells divided by a process called mitosis. The germ cell or reproductive cells divide by the process by the meiosis. The body cells or somatic cells divide by the mitosis, whereas these germ cells or reproductive cells divide by the process called meiosis. As of now, cell cycle. All cells are produced by pre-existing cells. Continuity of the life is depends upon cell division only, because continuity of the perpetuation by the cell division only, no? When there's cytoplasm as well as nucleoplasm divides means life will be continued, no, life will be multiplied. When cell divides, it synthesizes the various components including genetic material so that all material get duplicated, and duplicated material passes to the daughter cells. 1n, n plus n, 2n, no, these are continued. The orderly sequence of events by which cell duplicates its content and divides into two is termed as cell cycle. The sequence of the events that duplicates its content and divides into equal hours. So then this whole process is known as cell cycle. That's it. The above sequences is controlled or genetically controlled. The cell cycle can be defined as the entire sequence are happening from the end of the nuclear division to the beginning of the next. The cell cycle involves three following cycles. What are those? Chromosome cycle. In chromosome cycle, DNA synthesis alternates with the mitosis. During the DNA synthesis, the DNA double helical structure replicates and into two daughter DNA molecules. In mitosis, duplicated DNA molecules ultimately separated. Chromosome, these are two daughter DNA molecules. They are ultimately separated. Actually, these are double helical structure. They are twisted, no, one to another like this, and they are separated ultimately. Second one, cytoplasmic cycle. In this, cytokinesis alternates the cell growth. During a cytokinesis, cell as a whole divides into two. Usually, karyokinesis followed by the cytokinesis result in multinucleate cells. In polyembryony, we can be seen. Third one is centrosome cycle. Both the above the cycles, chromosome cycle and cytoplasmic cycle, in both centrosome be innerted and duplicated precisely in order to form two poles spindles either sides. So either side spindles fibers will be formed, no, when we see the metaphase. Third chromosome cycle forms third component of cell cycle. In 1953, Howard and Pilley both are divided the cell cycle in 4 stages. 4 stages means 5 stages. GO stage also will be, no, altogether 5. So let us see what are the stages in the cell cycle. First one, G1, gap 1 phase, gap 2 phase, synthesis, M phase, and GO phase. So these 5 stages we can be seen in cell cycle. G1 phase, G2 phase, S phase all together form a classical interphase. Cell cycle of somatic cells comprises essentially two phases. One is interphase, another mitosis. Cell, however, it spends the most of its lifetime in interphase only, in which cell doubles its size and duplicates precisely its chromosome complement. Means exact copy will be. The interface has is known today is a period of biosynthetic activity. Usually no cells cannot divide after birth. In human beings, what will be after 6 years? The no cell cannot divide. Human neuron, a interphase period lost entire life of the person. Usually in eukaryotes, cell division takes place by mitosis or meiosis. In mitosis, we have known double in size exactly. Precise chromosomes will be, 2 daughter chromosomes will be. In meiosis, 4 daughter will be, but chromosome complement will be reduced off to maintain the chromosome number constantly. The period of division represents complex series of stages by which cellular material divides between two daughter cells. Cell division only final phase. Before the cell enters the cell division, it was already duplicated its molecular components. Thus, cell division can be considered as final separation of already duplicated molecular units. As of now, interphase. So, interphase is called resting phase, but in fact, it is a great activity because there are important events or processes which are considered preparatory to the cell division takes place during the interphase. Means whatever we are required for the synthesis of DNA, this all preparation takes place actually in interphase only. Now let us see the general events in the interphase. Interphase characterized by following events or processes. What process are? Let us see. Nuclear envelope remains intact, means no change at all. The chromosomes occurs in diffused long coiled indistinct fiber chromatins. The DNA amount to become double. Replication of the DNA along with the synthesis of nuclear proteins such as histones takes place. Due to accumulation of the RNA protein and ribosomal RNA in the nucleolus, the nucleolus greatly increase in their size. In animal cells, duplication of centrioles takes place by outgrowth of daughter centrioles from the parent centrioles. Either sides, no, centrioles will be. So these generally these in plants spindle fibers will be. In animals, centrioles will be coupling structure which are above the nucleus. No, these which are right angle to the each other above the nucleus. Thus, an interface has two centrioles. Synthesis of energy-rich compounds takes place, which are required for the mitosis. And synthesis of proteins also takes place at the end of interphase. Once again, interphase can be divided into three phases: G1, S and G2. After M phase of the previous cell cycle, means before the daughter cells begins G1 phase of interphase of new cell cycle. G1 phase is a resting phase. It is called as first gap phase. Since no DNA synthesis takes place in this stage, that's why it is called gap phase. Currently, G1 phase is also called as first growth phase because many synthetic components which are required for the cell cycle reliably preparation period in this only. That's why it is called a first growth phase. Since it involves RNA, proteins and membranes, some of the membranes also prepares here only, which leads to the growth of the nucleus and cytoplasm of the each daughter cells towards the mature size. The chromosomes are fully extended all over the nucleolus. With light microscope, we cannot be seen like this. Not distinguishable as a discrete chromosomes. This is a time of resumption. Means start again normal cell metabolism do slow down during the previous cell cycle. G1 involves the transcription of three RNAs. One is rRNA, tRNA, and mRNA. rRNA, mRNA, and tRNA. RNA synthesis indicates that appearance of the nucleolus in the interphase nucleus. Regulatory proteins which control the events of the mitosis. Second one is enzymes. DNA polymerase. Enzymes are prepared, that is DNA polymerase. DNA polymerase is necessary for the DNA synthesis which are required for this reason. Third one, tubulin and mitotic apparatus proteins. G1 phase occupies 30 to 50% of time of cell cycle. S phase or synthetic phase. In this phase, duplication of the DNA and histone proteins are required in massive amounts immediately at the beginning of S period of DNA synthesis to provide new DNA and nucleosomes. The end of S phase. But DNA synthesis stops. In S phase only DNA replication takes place. No. Here will be stopped. Only RNA and synthesis of proteins will be continued because these two are required for the cell growth. In this phase, duplication of the DNA and histone proteins occurs at the beginning of S period of DNA synthesis to provide new DNA and nucleosomes. At the end of S phase, each chromosome has two DNA molecules and S phase occupies roughly 30 to 45% time of cell cycle. Second gap phase, G2, also called as second growth of phase, second gap phase, second resting phase. During G2 phase, synthesis of RNA and proteins continued, which are required for the cell growth. But DNA synthesis stops. In S phase only DNA replication takes place. No. Here will be stopped. Only RNA and synthesis of proteins will be continued because these two are required for the cell growth. This phase occupies 10 to 20% time of cell cycle. As G2 phase draws to close, the cell enters the M phase.